I'm happy to have this opportunity to speak to you today. I'm Masaki Goto, Senior Director of Biology Group of Axcelead Drug Discovery Partners, Inc. Today, I would like to talk about the importance of PK/PD strategy in drug discovery research. Before starting a seminar, I would like to explain this web seminar. This seminar will explain each part of drug discovery research in a series. Our experts in Axcelead will explain the important part of drug discovery research once every 2 months. Next time is a modeling and simulation, and the third time is a target product profile, TPP. I hope you look forward to this seminar. Now, let's get into the main subject and start my presentation. As you may know, what is a PK/PD? As you may know, I will review it. A PK is a time course of the drug concentration in the body, understanding of absorption, distribution, and metabolism, and the execution of the drugs. PD is a biochemical and physiological effect of drugs on the body and the understanding of the mechanism of action of the drug. A PK/PD analysis is to analyze the relationship between drug-induced pharmacological effect and drug concentration in the body. This is a textbook explanation. However, these contents are back to correctly understanding PK/PD and utilize it for drug discovery research. Therefore, I would like to explain in a little bit more detail. PK/PD and efficacy is important in drug discovery research. As you know, these are 2 major phases in clinical and clinical phase. Generally, each PD marker is considered in each phase, but it is important to establish PD marker and its evaluation system that can be used in clinical study from the early stage of drug discovery. Efficacy is also an important factor, but pursuing efficacy alone will not lead to success drug discovery research. Unless we conducted the drug discovery research with awareness of efficacy and PD markers that lead to efficacy, we cannot confirm what is happening and what is linked to the drug efficacy. In other words, this PD marker selection is very important, and we believe that conducting drug discovery research using PD marker will increase the probability of clinical success. Let's consider the importance of PK/PD in non-clinical drug discovery research. I think that you are proceeding your project, your research, while confirming the MOA, POM, and POC in animal model from the early stage of the in drug discovery. To prove this, we are evaluating the target engagement marker, downstream marker, and efficacy, and conducting our research while evaluating the relationship between these markers and PK. As I mentioned before, by checking these interactions, you can confirm what kind of the action is exerting the efficacy. In addition, the analysis of these PD marker and PK enable clinical PK prediction and estimation of the effective drug concentration. By clarifying the drug action based on the PK, we believe that exploration from animal to human will be possible. And next, I will explain a little bit more about these PD markers. Although these PD markers are important markers, they are often confused. Depending on the researcher, when we talk about the PD marker, the sense of the level of PD marker between them and I is often defined. However, I think that to organize a graphs, this point is the first step in understanding of PD markers. The target engagement marker is an index for confirming that this drug acts directly on the target molecule. And the downstream marker is an index for confirming did the drug change the function of the target molecule. POM can be confirmed by analyzing these markers with target exposure, that is the PK. In addition, after confirming this POM, we will confirm the POC for the first time by confirming whether the drug shows efficacy in endpoint. Next, let me explain these three markers in detail. The target engagement marker means confirming that the drug acts directly on the target. For example, binding to the target, intercellular signal directly under the target, or in the case of enzyme, enzymatic reaction. Downstream markers are often used to assess the cell function using the event that occur as a result of drug acting directly on the target. Downstream markers including proliferation, differentiation, production, secretion of the physiologically active substances, or migration and transformation of cells, and so on. In addition, endpoint is an index to show the drug efficacy, and included, for example, cardiac function, tumor formation, and behavior in cardiovascular, cancer, and the CNS area, respectively. It is really important to properly consider and grasp this series of the action and the effect. Next, I will explain by introducing the actual example from now on. In the first case, I will introduce the PDE10A. PDE10A is known as a target for the treatment of schizophrenia, which is a CNS disease. This is an enzyme that degrades intercellular cyclic nucleotide, cyclic MP and cyclic GMP, and is localized in the brain. When considering the target engagement, the better thing is the evaluation of the binding between the drug and the target. In other words, if you can confirm the binding of compound to the target, it is found that drug is working on a target. In addition, cyclic MP production can be used as parameter for the enzymatic reaction. It would be more encouraging if we could confirm the relationship between the indicator and the drug efficacy. In addition, it is important to use the same PD marker when considering the clinical development. But in the case of this target, we are proceeding with evaluation of the binding using PET ligand. In the evaluation binding of the target engagement markers, as shown here, the occupancy rate with the enzyme is calculated by competitive experiment with an excess radioactive tracer using the brain sample. In addition, since we observed that the production of cyclic AMP in which the enzyme reaction was inhibited by the binding of the compound was increased, we confirmed the relationship between this occupancy rate and the production of the cyclic AMP by compound in the rat brain in vivo. At the same time, hyperactivity, which is an indicator of the positive syndrome for symptoms of schizophrenia, was also significantly suppressed in correlation with the binding of PDE10A. Based on these results, it would be possible to calculate the binding rate required for clinical efficacy and proceed with development of this compound. Next, I will introduce genius kinase JAK. This JAK is located downstream of the cytokine receptors and is an important kinase for inflammation and immune responses. This enzyme is a target for the treatment of autoimmune disease such as rheumatoid arthritis. This JAK phosphorylated STAT downstream transcription factors to regulate gene expression such as cytokines. Here, the target engagement of interest is this phosphorylation STAT. It is very important to confirm the relationship with the drug efficacy using the level of STAT phosphorylation, p-STAT. In addition, the key point is to use the same PD marker when considering a clinical development. In the case of this target, we proceeded with clinical development using ex vivo evaluation in human form rat, like rat. As shown here, this is an ex vivo assay system. JAK inhibitor was administered to rat and blood was collected. Then the effect of STAT on phosphorylation was evaluated by flow cytometry. JAK inhibitor shows concentration-dependent suppression of STAT phosphorylation induced by cytokine. And this inhibition rate of STAT phosphorylation indicates the good correlation with the inhibition of the pore swelling induced by collagen in the rat arthritis model. This clinical development of JAK inhibitor was being carried out using this relationship. This is the actual clinical data. As shown here, the effective dose of the each JAK inhibitor is determined from the IC50 value for STAT phosphorylation in ex vivo studies in healthy subjects and their blood concentration of JAK inhibitor. And you can see the good correlation between the p-STAT and the disease activity score indicates the therapeutic effect on rheumatoid arthritis patient, like non-clinical cases. This analysis of PD marker is also important in clinical development. Furthermore, PD markers should be selected according to target and disease. Some of the data presented today, including these, are not ours, but except for the clinical data, we have experience in everything, so we can carry out them in-house. The phosphorylation of STAT in JAK ex vivo studies was evaluated using a flow cytometer with 5 lasers. As for the PDE10A inhibitor mentioned earlier, the release of dopamine in the brain can be measured as a PD marker as shown here. This dopamine release can be detected by this microdialysis system. By implanting a special probe in specific part of the brain, this system can measure the neurotransmitter with anesthesia. Furthermore, this system can also measure extracellular compound concentration in the brain. So this system can detect PK/PD in the brain at the same time. Finally, I would like to summarize this seminar. In the drug discovery research, it is very important to establish the PD marker and its evaluation system that can be used in clinical study, considering POC and POM from the early stage of drug discovery. And it is also important to select an appropriate PD marker in consideration of the relationship with target engagement marker, downstream marker, and efficacy. Today's seminar was a little bit conceptual, but let me explain the importance of PK/PD efficacy. However, drug discovery research will not succeed unless not only this PK/PD efficacy, but also related parts such as measuring chemistry and modeling and simulation and so on are connected. We have many specialists in these parts, and we hope that they will be useful in promoting your project and solving your issue in drug discovery research. Why don't you join us in drug discovery research? That's all my presentation. Thank you for your kind attention.